Okay, my good friends, this is Roger once again, and I'm going to go over how this biology turns into what we see as geology. Now, what this is from Tyson Carlson, very good guy, very knowledgeable about this stuff, studies it very well now. I've been working with him, I don't know, got to be a couple of years, and I discovered the mud fossils in 2012. And I have small ones here. They're big, but because that's a tendon ball. You see that? That's a tendon ball right there. We're going to see one right here, and they come down on straps. That was another one that came down further. You see that's the way they come, and there's a ton of them, and they're all over the place here. And what they do is they hold tissue in. They lock it in, and then the tissues can move around while the tendon balls are the anchors. Now, they're everywhere in the body. I mean, they're literally everywhere. And in us, they're so small, we really can't really see them. But in these big creatures, they're huge. Now, what I just showed you was a tendon ball here that solidified in a certain way that the mineralization there stayed. You see it? How those white spots are in there? They would call that basalt. Now, here's another one. They're petrified in a different place. You see all those holes? Right? Instead of having the mineralization lock in there, this particular one was in different chemistry. And what it is it's a long time in water, aqueous solutions. And in this particular one, whatever was in that water attached to the molecules that were in that little spots, the little holes there, the white spots. That there's a certain mineral, and I don't know exactly what it is, but is that mineral in there. That other one was in the chemistry that it ate the mineral and washed it away. This is all due to long time watery conditions, and there's saltwater conditions and there's freshwater conditions. And in my case, my stuff was in salt water originally, then it must have been flushed with fresh water after it went through the preservation process. Then it had to be extremely long duration of fresh water to take away all the invasive molecules that were the ones that were causing things to convert. And like this goose head. You see the feather patterns? See the feathers on his head? It's a goose. These things, people haven't noticed this. Nobody's ever seen it. It's hard to believe, but it's true. And every single thing there is biology. And I'm going to tell you what. Tyson's thinking this is deep in here. We go back and forth about how we see things because, nobody knows anything about this. We don't know what kind of biology this is. This could be a fingertip. I'm serious. This could literally be a fingertip in the creature. It's absolutely stunning what he has, and he's understanding the chemistry. He's going out looking at it. Every day he's going deeper and deeper into this. And because there's so much to learn, and we can make mistakes. We could be right one day, we could be wrong the next day. So we go back and forth at this, and this is something that everybody that is interested is going to have to dig into deeply. You can't just look at, oh yeah, and then that's the end of it. No, because when I first discovered this, I only had these small ones. When I say small, I have giants which are a couple 100 feet tall, but there's nothing like this. So, my looking at it, I never saw tendon balls. Like none of my stuff I can see in this kind of detail. I mean, this is the best site in the world right now. I think. And I just don't have any clue how big this thing was, but I can tell you what, the cell membranes, the bilipid cell membranes, which I'll show you as we go through this, because we're going to go into the chemistry deep. It's time to do it, because now I can see it. I can really see it now. Before, I could see this, and I could see that that was black in there, and I could see there was a little bit of bone, and I could see there where the ligaments attached, and I could see where the arteries and the veins and so forth were. But this is a whole another level of seeing. This is like having freaking electron microscope when I just had my plain eyeballs before. So we're going to go deep, and we're going to understand feldspar, which everything they say is feldspar. Oh, that's just feldspar. No, it's not. That means it's tissue. This is a dinosaur egg or some kind of egg. And that, they say, oh, it's just feldspar. No, it's not. It's biological tissue. That is the exact same stuff, biological tissue. And it's feathers, it's skin, it's interstitium, it's all of the things. Like this is a lung, it's the same stuff. It's the same stuff. It's the biology of life. It's the fibers of life. Anyway, we're going to go in there deep. So get ready. Okay, my friends, now you saw the giant stuff we're talking about. And this stuff was all written about in the ancient texts, including the Bible. And that's why it was written about gigantic dragons and gigantic fish, which that's what it is. It's in the desert in Morocco. You can easily go there and look at it and watch what happened in the, and it couldn't be that long ago because these are on the surface of the earth. This giant dragon here spit this stuff out to try to kill that giant fish. Now you can see what it is here in the desert. It is, whatever this is, that was a combustible, nasty, noxious, explosive substance that funneled down on that fish to try to kill it. And it certainly did damage it. Now, that is the dragon, that is its throat. This is not something that can be dismissed unless you're a professor or a geologist. Then you can dismiss it and say, no, I read in a book that that's not supposed to be what you're saying it is, even though there's no way you can dismiss it. No. This is, I think it's 1,100 miles long. So Tyson's stuff is, it is what it is. It's time to pay attention to this. And the universities have turned their backs on reality, 100%. Yale knows every bit about what I've been talking about. I've sent them all my DNA reports and CAT scans, and it's the same with Harvard and all of them. And they refuse to engage because they know now that they have been exposed for not being correct. And they won't engage. I don't think that's right, but that's what we're up against. So I'm going to be showing you this stuff, and just stay with me and engage in reality. That's all. So let's get back to how this stuff converts into preservation, because here you have gigantic creatures, yes, but Tyson's in a situation where it's literally eating into it. I mean, he goes back there daily, and it's different. It's absolutely incredible what we can learn on his site. Now, I also have someone, Tish Egerton. Now, let me just show you something. I discovered the no-toes when I first discovered my stuff. I discovered everything basically at the same time. And, giants, no-toes, dragons, I mean, everything. So it was just like, wow. But Tish Egerton saw, I put a video up, says you can find this stuff in 15 minutes, guaranteed. She goes out and in 15 minutes, I'm serious, I'm not kidding you, she finds a village. And she's got the same situation. Mine, everything I have is literally perfect. It's almost like it was never even, I mean, it's absolutely flawless. You know what I'm saying? I mean, it's not deteriorated. Now, Tyson's stuff, it's eating right in. Tish's stuff is in like layers of degradation that give us we can look into it just like we're looking into Tyson's stuff. Let me just show you quick Tish's stuff, and then we'll go on back to Tyson's area. All right, you saw that I have this perfect no-toe now. And the toes are inside of them. Now, these are Tish's. Well, she's got a ton of these, so this is just part of it. But they would have had the same enclosed toes, but they're eroded away. The toes are in there. Now, she also had, she got a ton of stuff. Let's see. This is a leg. All right, so there's the leg coming down. That is like tendinous material. These are springs. Now I'm going to show you what they look like in. She's got another one that's eroded even more than this, and we can see the extreme details. And here it is right here. Now, this right here is the heel bone. There's a strap, a tendon strap that runs right up here to the bone that comes down and sits in this saddle, which is your tibia. All right, and that goes right up like this and attaches to the tibia. And I'm going to show you that anatomically in a minute. Now, in us, we have bones that run down here and tendons that run down to the toes. In this is literally constructed. And I'm not kidding you. You see up here? You see that? There's a little spring and a latch, and it goes up through here, and it comes back over here, and it comes down to here. All right, somehow this thing wraps around here and ends up getting back to here. Because somebody else sent me one that is, this part is even eroded. And you can see there's like a circular spring around here, and this wire goes right up through that spring. Very unbelievable. Now, so how does this actually work as a springy foot? Here's how it works. The toes are out here, and they're just little bumpy toes. And when you step on them, they move up this way. Now, whoops, hold on. Okay, my friends, I want you to know that I study the anatomy that has turned into rock. That's a lung. Any doctor, any anatomist can tell you that is a lung, and that is the pleura of the lung, which is a substance that is created by the body using collagens and fibers and so forth. Now that is an opalized heart, a human heart, and that is, you can see that the tissues are extremely specific. That's why these certain colors are only in certain areas. And the reason they're that way is because of the chemistry. And the chemistry is only that way because of the tissues. And the tissue is only that way because it has a specific job. It's either springy or it's rigid or that type of thing. Now, I am deep into this. Rocks, this is a goose's head. And that's the feather patterns on his head. All right, that's his eye, and that's a beak, and that is a goose's head. And that's his neck, and he died laying like this. This is a human lung. It's been DNA tested, CAT scanned, anatomist verified. That's the depression of the heart. It's a left human lung. This is the pleura. This is a new species that we are looking through now, which is called no-toes. I've named them. And we have these in enormous quantities. And there is so much that is not understood. And I came into all this because I discovered these, and I named them mudfossils, which are ancient human and other types of creatures that we never knew about. And the bones that we thought you had to be white, shiny looking bones, no, these turned into literally into mud and dirt, actually, which we would call mud and dirt and just rocks, but they're actually superbly preserved because of a process that happens in wet, salty conditions. And then mine have turned into very good specimens because they must have been flushed with clean, unsalty water shortly after being gone through the soaking preservation process, which the salts preserve it, because there's a lot of invasion, and then you get rid of all of the extra salt in fresh water. I've made mud fossils. And it's not hard to make. It doesn't take long at all. It only takes a year or so. And you end up with just what you're seeing here. You end up with the entire product. Now, science right now and the they're not looking into things. They're looking at the old books. They're talking about geology is just all, none of this stuff is real. They won't pay attention to reality. And the same things has happened in the medical field. And it's kind of disgraceful, really, that they have to say the things that they were told to say, or they'll actually be destroyed in their positions. That's the key now. The academic society has become so rigidified, and you cannot step outside your boundaries. You have to defer to someone else's authority, and they only have that authority because they say they have read enough books that they are now fully invested in that knowledge, which is not, it's not correct. It's just not right. So you read all the books you want. If they're not right, you still have no knowledge. And that's the case now, because none of this stuff is accepted by any university at the moment. And they won't discuss it because they have no way to discuss it and to win. I have the truth and the facts and the evidence, and they have nothing to support anything. I have the DNA reports. I have the CAT scans. I have the specimens. I have everything that they do not have, and all they have is denial, and they're just hiding. This lung was DNA certified. I have giant human beings that were CAT scan DNA certified. Here's a fingertip. That is a fingertip. A fingertip. CAT scanned, DNA tested, certified in every single way. All right, time for them to change their tune. Okay, now, I get stuff from all over the world, and this came from Gary Evans, who got this out of a salt flat. I believe he's in England, somewhere on the coast of England, and then he broke this open. Here's what he found. All right, here's what he found when he broke it open. It's a lung, quite obviously. Now, there's two different, you see the reddish area here, and you see the real blackish area here. That is literally how your lung transfers oxygenated blood from deoxygenated blood. One's black and one is red in mudfossils. In us, it's more or less bluish and red. Now, you can obviously see the blood vessels are, I mean, just absolutely phenomenally intact. And the blood is here, and it's just exactly what it is. Now, so he takes this out of the mud. Now, here's where I need to explain to you why my mud fossils are literally flawless and perfect. And this goose head didn't rot away. All right, it stayed, feathers and everything. Now, why mine? And let me show you what happens to his, because this was found in a salt flat. Now, here's the conditions that happens. You have a lung. This is a lung too. Now, but this one was in different chemistry. Now, this is in salts. Salts in your body, you live in 7.3 pH basically, and if you stayed in 7.3 pH after you died and you were encapsulated in wet, fine, platy polar silicates, which is extremely fine mud, where no oxygen could get in there, this is what would happen to you. You would become preserved, basically like pickled, from the invasive chemistry that is basically your salty, same stuff you're in. Your body doesn't even know you're dead. That thing doesn't even know it's dead. Now, however, once you remove this chemistry, this pH condition, and then you let it dry out, now you got a problem. All right, you saw how perfect that lung was before. This is what happens once it starts to dry out, when oxygen hits it. Now, mine is another lung just like this, only mine was preserved, I'm sure, well, I'm positive, in that salty condition, which gave it the preservation asset. And then it was completely flushed in extremely, pH neutral, probably, somewhere in that area, or low pH. I'm not sure exactly, but it was not salty anymore, and it ate the salts out of there and preserved the preserved tissues. So the tissues just stayed the way they were after they were preserved by the salts. That's my take on this. And see, that's what happened to Gary's. It just deteriorated. It turns into mud. That's how you get mud. Flesh and organs that were rotted away, literally, that's what mud is, and dirt. And then you've got other ones like this. If they're in this condition, this is the key. It's all about conditions. Where did they find it? How long was it in the salt? How long was it in the other conditions? How long was it dry? Tons of conditions. Look at it. That's a toe. That's a toe. All right, this is invaded with all kind of different transition metals. Why is that? Because it was in blood. It's like there's blood all over. See, this was very similar to what happens with opals. Only opals, it's a little different chemistry. And then you get petrified wood, agatized stuff. There's so much to this that it needs a lot of research. And I've done a lot of research. I've done a hell of a lot of research on this. I understand this stuff pretty good. I'm probably as good as anybody. Well, better than I can make that statement. I understand it better than anybody else. I discovered it. I did all the research on it. Tyson's coming along very nicely, but that's about it. And I have some other people that are looking into it, but not like the depth that we are. And that's what you got to get in very, very deep into the chemistry, into the biology, into the anatomy, into the degradation, and how everything erodes and destructs. And really now I'm into the electron flood theory, which is everything about chemistry. It's all wrong. Chemistry right now is the end result, yes, I can agree with that, how they understand how all that works. But from the beginning, from the nucleus, it's totally not understood anymore. The nucleus is all particles that are electrons, but the electrons are dipoles, a strong and a weak force. Exactly what they've seen, but they don't understand. They think there's still gigantic neutrons and protons in the center. Absolutely not. They're just globs of electrons, and the explosive part of the electron, which is the fermion, which is the strong force, will always be on the outside as a crust. Therefore, the other electrons that want to get into the positiveness in the center, they are held at bay, and it's called the quantum. I've got all this stuff very, 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 very well shown, and it can't be unshown. It can only be ignored, which is exactly what's happened. Now, I don't want to go long on this, but I hope you understand the things that we are going to explore are going to be what I would consider to be reality. If somebody can come up with any scientific material objections to what I present, I'd like to know. But just to hear that, oh no, that's a schist. Oh no, that's igneous rock. Oh, that's sedimentary layering from, that's lava. That's absolutely not. No, those things were all wrong, and we showed this in extreme details. So, like I said, I don't want to go too long on this one, but we're going to go back at it, and the next video, I'm going to do a series of them. I'm going to go through Tyson's stuff, because his stuff is just phenomenal. It's, we can look at it and walk on it, because he's jumping up and down on tendon balls. He could do jumping jacks inside of veins. I mean, it's just unbelievable. So now, and the chemistry is there. There's no mysteries here. The mysteries are gone now. Now it's time to, what kind of creature was it? Why are these tendon balls different? What kind of tendon ball is in what different place in what anatomy? Because there appears there's ones different styles that are in the interstitial areas and your skin, fibers and all kinds of things we're learning now. I think it's really fun to go deep. Some people just want to go on the surface. That's fine. But if you want to go deep, stick around.